on the toxic impacts of alpha-synuclein. That is protein about 140 amino acid sequences, and it is deeply associated with the synaptic vesicles. The presynaptic localization of this protein suggests that this protein especially takes part in the neurotransmitter release. However, aggregation, mutations, and other changes in the alpha-synuclein that is also found in the Parkinson's disease. And yet, there is no clue what is the specific function of the alpha-synuclein and what lead this protein to the aggregation. So, I'm going to present my experimental finding, few experimental findings on how this mutated form of the alpha-synuclein that is mainly seen in the familial Parkinson's disease patients, and how when this protein is overexpressed in the neurons, then how this protein is toxic to the Parkinson's disease patients. I have done the experiment that is based on the two mutations that is on the N-terminal sequence of the alpha-synuclein protein. You know, N-terminal sequence of the protein consists mainly of the amphiphilic proteins that is very deeply bound to the phospholipid layer of the neurons. Consists of about 1 to 60 amino acid sequences. Then from 60 to 95 sequences, that is NAC domain, that is non-amylite component, that is responsible for the beta structure of the protein, and then the highly acidic C-terminal domain, that is very negative and very acidic in nature. This protein actually, when it is present in a native form, in our physiological conditions, this protein is present in the form of an alpha helical structure that is known as native protein. Once, due to the age or due to certain conditions, when this protein becomes aberrant, misfolded, then that misfolded protein that gives a highly misfolded intermediate in the form of oligomers and the fibrillar protein. And this fibrillar protein actually, that is the disease-causing protein that is found in familial as well as non-familial Parkinson's disease patients. So we have taken the verified recombinant alpha-synuclein protein and we made it the size of the oligomeric proteins by means of shaking and by means of incubating, and then continuously shaking at 1000 RPM for 7 days, and then stored at minus 80 degrees centigrade. And you can very well see here the oligomeric alpha-synuclein protein that is localized at about 30 to 40, about 30 to 35 kilodaltons. We wanted to know that how this alpha-synuclein protein, how actually it interacts with the mitochondria. And for that purpose, we incubated the aggregated protein that we have made aggregation by shaking it. That aggregated protein was incubated with the mitochondria, and we found the same aggregated form of the protein that bonds very well with the mitochondria also. So, what lead to the aggregation and how it causes the neurodegenerative disease? There are some genetic factors as well as environmental factors, both contributes to the disease process. Basically, the mutations, two mutations that I told you, at alanine 53 to 309, as well as alanine at 30 position to proline. These two mutations that are very well reported in the familial Parkinson's disease patients. Both of these, like the overexpression of the protein as well as the mutation of the protein that causes the protein aggregation and that is actually the main cause of the neurodegeneration. Here I have presented how this alpha-synuclein is toxic. Linear alpha-synuclein that is present in native form also, when it is overexpressed, then that also causes the neurodegeneration by means of cytochrome C release from the mitochondria and ultimately apoptosis. The protofibrils that develops during the misfolding of the protein that causes the increase in the cytosolic dopamine and that produces the reactive oxygen species. Then the fibrils, they are actually the main toxic species of the alpha-synuclein, and that causes the inflammation, microglia activation, also inhibits the natural alpha-synuclein functions and also produces a lot of reactive oxygen species. We wanted to see how this alpha-synuclein, how it interacts with the mitochondria and produces the reactive oxygen species. So this is a model where I have shown that this protein interacts with the outer mitochondrial membrane. After interacting with the outer mitochondrial membrane, that goes inside the mitochondria and causes the release of the cytochrome C that activates the caspase activity, and the cell death is caused by means of the apoptosis. So we have overexpressed this alpha-synuclein along with the mutant species of the alpha-synuclein, and by means of alpha-synuclein antibody, we have localized the presence of the alpha-synuclein inside the mitochondria. By means of electron microscopy, we were able to show that this alpha-synuclein goes inside the mitochondria and is very well localized inside the mitochondria also, that is the powerhouse of the cell. We have seen that this alpha-synuclein wild type as well as both mutants A53T and A30P, both mutants as well as the wild type that causes the aggregation of the protein inside the cells. This is the picture shows the SHSY cells, and in the middle you will have the mitochondria picture that is stained with the mitotracer red. Then the lower one is the merged picture where we can very well see the localization of the proteins in the mitochondria. By means of several Western blot, we have seen that the mitochondria has both the types of proteins, that is the linear form of the protein, non-oligomeric, that is monomeric forms, as well as the oligomers. Both types of proteins, they are bound with the mitochondria. We have done several types of experiments in the mitochondrial membrane, whether that alpha-synuclein binds to the mitochondria or it is remain in the cytosol or remain in the supernatant only. So we have found that the alpha-synuclein protein that is very well localized and binds to the mitochondrial membrane, probably because of its amphiphilic N-terminal end of the protein. Then we have tried to incubate the mitochondria, the lower one on the right side. Incubate the mitochondria with the 10 microgram and 1 microgram of the recombinant alpha-synuclein. And we have seen that the alpha-synuclein is very well binds to the mitochondrial membrane. We have then, by means of confocal microscopy, we have seen the presence of reactive oxygen species that is very well elevated in the wild type as well as both mutants, and that activation that is very well localized in the mitochondria itself. The last row, that is the merged picture of the mitochondria as well as the cytosol, so that here it is very well seen that the reactive oxygen species is elevated in the mitochondria itself. Then by means of tyrosine hydroxylase antibody, we have done immunostaining of the cells. They are expressed by the wild type as well as the mutant form of the alpha-synuclein. And here also we have seen that a lot of tyrosine nitration that is caused by all the types of alpha-synuclein wild type when they are overexpressed, as well as the both mutants A53T and A30P. The mitochondrial transmembrane potential was lowered. The lower portion of all the 4 blocks of the slides that shows that FCCP causes the decrease of the transmembrane potential, and the upper one that shows the transmembrane potential in the wild type and both mutant forms of the alpha-synuclein. The respiration, mitochondrial respiration was also decreased very much. So the question is what we can do about it to make sure the neurons, they are not affected by the aberrant form or misfolded form of the alpha-synuclein. So we may have several chaperones that can prevent the toxic alpha-synuclein. That is the one way. The second way is that we can develop the antibodies against the misfolded alpha-synuclein, because if we develop the antibody against the native form of the alpha-synuclein, then that will inhibit the natural function of the alpha-synuclein also. That may be involved in the neurotransmitter release specifically. Then the third option is to find small molecules that can prevent the misfolding and reduce the toxic effects of the alpha-synuclein. So for this purpose, we have taken the combination of 3 very important bioactive components: pyrethrin, tinocardicide, and acetylglucoside. All the 3 combinations, they were present in the equal amount, and the dose were 500 milligram in the animal, as well as in the cell 20 milligram in the cell. We have also done. Formed the alpha-synuclein, aggregated alpha-synuclein by means of several processes like keeping for 7 days in the under the incubation period, and then we have injected the same alpha-synuclein, misfolded form of the alpha-synuclein, into the striatum of the mice, and we have seen that decreases the swim score of the mice. Then we have taken the striatal cytosolic fraction, and we have analyzed the presence of the alpha-synuclein, and that shows that these bioactive components, when they are pre-injected, when they are supplemented to the mice before the intrastriatal injection of the recombinant alpha-synuclein, then that decreases the protein aggregation in the striatum. We have measured the same superoxide anion production in the striatal fraction after after giving the intrastriatal injection of the recombinant alpha-synuclein and the bioactive component. The top one, you can see that the bioactive component, they decreases the superoxide anion production also. We have taken the sirtuins. SIRT3 is the mitochondrial deacetylase, and the main function of the sirtuins in the mitochondria is that it controls all the bioenergetics process and regulate the mitochondrial functions. So therefore we thought that co-expression of the SIRT3 might decrease the toxic function of the alpha-synuclein. So first we localized the presence of the SIRT3 in the mitochondria itself, and we have seen that SIRT3 is very well present in the mitochondria. The upper blocks, they are from the electron microscopy, and the lower one is the immunostaining by anti-SIRT3 antibody. The co-transfection of SIRT3 with alpha-synuclein causes the reduction in the reactive oxygen species in the mitochondria. Here all you can see the control as well as wild type and both the mutants where reactive oxygen species was lowered. Then we have seen its effect in the mitochondrial membrane potential also, and we have found that the membrane potential was significantly maintained. So, in conclusion, we can say that alpha-synuclein interacts with the mitochondria and impairs the mitochondrial function. The bioactive component that we have used that was very effective in decreasing the toxic effects of the alpha-synuclein, as well as it causes lowering the aggregation process of the protein as well. Then 33 overexpression, because it controls the mitochondrial bioenergetics, so therefore it was able to restore the mitochondrial function that was demolished by the alpha-synuclein overexpression as well as the mutant form of the alpha-synuclein. Thank you so much. Any questions, please?
Yes. Yeah, go ahead.
Thank you for your lecture. Apart from the genetic predisposition, do you think that this virus is the EG is another source?
Yes. Actually, the familial alpha-synuclein pathology occurs only in a few cases only. 1%, not more than 1%. So therefore, it was thought that what is the toxic species actually that is really causing the pathogenesis in the Parkinson's disease patients. And it was found that the protein, misfolded protein, mainly fibrillar form of the protein, they are the main cause of the Parkinson's disease. That's the main pathology that has seen. But yet, the functions of the alpha-synuclein inside the neuron is not yet known. Nobody knows the function, but people, they predict that that might be involved in the neurotransmitter release.
That was my question, actually. We still don't know the real function, the role of this protein in the membrane, in the mitochondrial membrane.
Yeah.
Can we find it in the cell membrane?
Yes. It bonds to the cell membrane as well. Yeah.
Okay.
It is actually the N-terminal end of the alpha-synuclein that is amphiphilic in nature. And that can very easily, it can interact with any membrane.
So
I have seen only in the case of the mitochondrial membrane that it really interacts with the mitochondrial membrane, bonds to the mitochondrial membrane very effectively.
now, you mentioned that this protein is folding by chaperones,
Yes.
but chaperones mostly do their work not in the membrane but intracellular.
Yes.
Intracellular.
Yes.
Because most of all membrane protein are already folded.
Yes.
And so the problem is probably
Folding.
either in the matrix or in the mitochondria or either in the intracellular
Yeah.
matrix.
Yeah, that's
Okay. Well,
Yes.
yes.
Do bioactive compounds have any effect on the proteasome in dealing with the misfolded proteins?
Yes, they were able to decrease the aggregation. We have studied the aggregation and binding with the membranes. So they have decreased very effectively. They have decreased the aggregation of the proteins.
By the way, the sirtuin family is a large family. If we would like to enjoy the benefit of sirtuin, you should drink some red wine once a day. Yeah, I'm not joking.
Yeah, sure.
I think the group who found the sirtuin family will get the Nobel in the next few years. We will hear about them. It's a group from California. Yeah. Okay, thank you.
Thank you so much.
Well, next speaker is Jimenez, vestibular function. I'm getting dizzy from the